Hey everyone, I'm Michael. I'm a PhD student in the Culhane lab in Limerick, and as Eric introduced, I'll be presenting demuxSNP. And what does Demuxlet do? It's for supervised demultiplexing of single cell RNA sequencing. I will use both the cell hashing data and SNPs. So, in terms of what we'll go through today, you can install it from a couple of different places. So there is workshop material up on the Galaxy instance. We've a pretty short time here today, so if you want, you can go through it now, or else it's there when you go home later or next week or whenever you're interested. As well as that, you can take it on Docker if you want. And for some reason, if neither of those work for you, you can probably install it as a package from GitHub as well. So prerequisites-wise, people are familiar with our Bioconductor. That's obviously helpful as well if you're working in single cell data and have previous demultiplexing experience, then it's probably the place for you. Syllabus-wise, so I've it broken up into 3 different sections. So as well as the actual package demo part, I've thrown in some added bonus material. So first of all, there'll be a little like intro to demultiplexing. I'll show what the hashing data looks like, what the SNPs data looks like. Then I'll do like a very short benchmarking just to motivate what our package does. And finally, there's the actual package demo itself. So if you have any questions, feel free to shout out. I'll like do a quick stop after each of the 3 like subparts to see if anyone has any questions, and we can go from there. So the first 2 I'll go through on the GitHub pages just to keep things moving. Then hopefully for the last section, package demo, hopefully some live coding if everything works out okay. Great. I shall start off my Galaxy instance so that that's loading in the background. So given the conference we're at, I presume that most people here are already familiar with single cell and all the wonderful things that we can do with it. So I'll get straight to the multiplexing things. So here we'll take a step back a minute and look at how we get the data. So in droplet-based methods like 10x genomics, we have this droplet encapsulation step, and this allows us to measure at a single cell level. And so what we get is hopefully one cell, one barcode encapsulated in a droplet. Ideally, we'll get 1:1:1. But we don't always get this, it's stochastic, right? So ideally, we'll get a singlet, 1 cell, 1 barcode, 1 droplet. But we can also get an empty droplet where we've captured no cells, or a doublet or multiplet where we've captured multiplet cells. And these are problematic because we no longer have single cells. So we need to remove these. Now, if you just load 1 sample per lane of your sequencing run, you're not fully utilizing the capacity of your lane, right? And so we want to maximize this to reduce estimates and costs. So when we multiplex, then we take cells from multiple different biological samples, pool them together prior to being sequenced, and then they're all sequenced together on the same lane. So now instead of paying for multiple lanes for each sample, we're now just paying for a single lane. So now we're fully maximizing the capacity of the lane. However, we are getting some more doublets. So as before, we can get singlets, empty droplets. We can now get 2 types of doublets or multiplets, where we either can get 2 or more cells from the same sample, called single sample multiplet, or 2 or more cells from different samples, called multi-sample multiplets. And the more cells we load, the more doublets we'll get. So with demultiplexing methods, we can identify these multi-sample multiplets. So the first method, really commonly used if people are working on single cell data, they've probably come across this before. So it's cell hashing. So prior to the cells being pooled, each batch or each group of cells is labeled with a unique barcode antibody, which is conjugated to the cells. However, when we mix all the cells together, when they're pooled, we get non-specific binding, right? So some of the antibodies are going to come off and they're going to reattach to a different cell. And we get this between all the different groups. And that gives us this very distinct bimodal distribution that we see here just in the bottom left. So we'll get an upper signal, and these are cells that were originally stained with the antibody. And we also get the signal. So these are cells that kind of picked up a floating antibody, but it's not the barcode that it was initially stained with. So if you look at what this looks like in terms of on each individual cell, so here we've 4 different groups pooled together, hashtag 1, 2, 3, 4. So in the top right here, we have an example of a singlet. We can quite clearly see it's from the hashtag 2 group, right? There's a much stronger signal in hashtag 2 and a little bit of non-specific binding in 1, 3, 4. A doublet, then we'd expect to see a strong signal in 2 groups, or maybe 2 or more groups, and a lower background in the rest, as we see here in the center right. However, we can also have what we'll call a hashing negative. So in this case, we have a cell encapsulated by the droplet. RNA quality is good, but for some reason, either experimentally or computationally, we're unable to determine which of the groups it came from. These are problematic then, because it means we're not able to assign these because of hashing quality, not because of the RNA quality. So now let's have a look at the data. So there's a couple of ways that we can plot the hashing data. So first, basically what we get out is this counts matrix across each of the 4 hashtags, which is what they were stained with for our 4 groups here. So we see the red here is where we have high expression. So for example, cells around my mouse here, we'd expect to see from the top group here. If we see the red in 2 or more groups, it's indicative of a doublet. And if we see mainly blue across the column, that'll be indicative of a hashing negative. And I'll show a kind of clear example of that later on. So there's a couple of different ways we can visualize this. We can also visualize like per cell, as we saw previously. So this is some data from the SciFuse package, and it shows possibly like the best I've ever seen come out of a hashing experiment. So here we've like next to no background staining, and so we can very clearly see which one it came from. However, this is like an absolutely ideal scenario. Often we'll get a lot more noise, a lot more background staining, which makes the classification more different. And hopefully I'll get to show some of that later on. So we can also show a histogram of the expression of a particular hashtag across all the cells. And that again gives us this distinct bimodal distribution where you have an upper signal and lower noise. And there are some qualities of this which we'll explore later on, which determine how well we can use this to demultiplex. We can also do some principal components analysis, and some papers have kind of used this as like a heuristic for the data quality. And basically how this can be interpreted is the easier it is to split out the different groups in PCA, probably the stronger the signal of the HTO tags, and better quality hashing data is. Now here we can see we've almost split out the 4 groups. However, we can see, I think it's hashtag 2 and 4 just kind of superimposed on each other down here. However, if we then go on and extend this to t-SNE, we can see the 4 groups split out. And as well, we can see some kind of doublets falling in between the 2 groups. So next, the second kind of class of algorithms is based on SNPs. So these use natural genetic variation between genetically distinct samples to figure out which cell came from which sample. So how does this look? This is like toy example, not representative of what we'll get in reality. So in theory, you'd see some SNPs in some patients. Here we've simulated 20 SNPs for 50 patients in each group. In your single cell RNA-seq experiment, these all get jumbled up. And then going down, you have to try and figure out which came from which. Obviously, this is very clean data, very easy to cluster, and we can reproduce our samples very easily. So in practice, much, much harder to do. So first off, we're looking at single cell RNA sequencing, and short reads. So this creates a lot of problems. This kind of experiment isn't designed for good SNP calling. However, we hope here to just be able to get enough SNP information to figure out which cells or which group each cell came from. So this is really, really hard to decipher, right? Very sparse and not a lot of signal. So let's filter this down a bit to remove some of the kind of missing rows. So to explain what each color means, so we can either have no reads to support the SNP, SNP supported in the alternative, the reference, or in both. And I think if you start to look at this long enough, you can kind of start to pick out patterns, but it's much less interpretable than the hashing data. However, we have algorithms like Supercell and Vireo, which are designed to operate on this data. Again, to make it kind of a little bit more easy to see, in my opinion, we can recolor this to just look at, is the SNP absent, present, or we don't have information for it in the alternative, which is in our data. And this is the kind of the color scheme and representation that I'll use for the rest of the presentation. Everyone with me so far? Great. So next, I'm going to look at some very short simulated benchmarking of one hashing-based algorithm. So what we're going to look at here is some simulated data. So how we generate it is, first of all, so we're drawing from either a background or a signal. So you start off with this binary matrix. So if it's a 1, it indicates I want this cell to be from single or 0 from the background. So we can see on the far left here where we've red, so they're going to be doublets, right? We've signaled in both, and we're just looking at 2 groups here. So going across the rest, so this here would be signal in #1 for these cells. Here, signal for the #2 in these cells, and over on the far right, we have negatives. So we're drawing from the background from these. So we then draw from negative binomial distribution. We can supply the mean and size parameters for this, and we can adjust these as necessary. We can also specify how many cells we want in each group, how many doublets, how many negatives. So this is just a toy example to illustrate how we generate the data. Again, obviously, it's very nice to use real data for benchmarking. However, when we're looking at demultiplexing, it can be very hard to get a good ground truth. So here we're just making use of some simulations so that we're pretty sure what it should be. And again, we can see the bimodal distributions for these. So first of all, let's simulate some really, really high quality data. So here we've 4 groups, #1, 2, 3, 4. And as well as that, we've simulated some doublets and negatives to add into the mix. So this is high quality data, and we can be pretty sure of this because we see this really, really clear distinction between signal and the background in each of the 4 groups. So when we apply H2 Demux, a popular demultiplexing algorithm to this data, we see very, very good results. So in #1, 2, 3, 4, and in the negative groups, we see exceptionally high precision and recall with only slightly lower precision in the doublet group. As we expect, regardless, most algorithms are going to perform really, really well on this data because we have a very clear distinction between the signal and the noise. Now, let's look at some poor quality data. So here we've changed a couple of things, right? First of all, we've overall reduced the signal to noise ratio. So we're starting to see some overlap between the signal and the background. As well as that, we've varied the number of cells in each group. And as well as that, we're kind of changing the mean, so it's occurring at a slightly different spot in each group. And these are representative of some features that we see on some of the real data sets that we've come across. And so again, we apply H2 Demux, and we see much poor performance overall. So if we look at #3 here, which was a very large group, but low signal to noise ratio. So the recall here is particularly low. And we see some poor performance across the other groups as well. So this is just one algorithm. We expect slightly different algorithms to perform slightly differently on various reductions in data quality. However, overall reduction in hashing quality, the algorithms are going to perform worse. So where are some of these cells going? So for example, if we look here at this #3, which is one of the large groups we had, we see an absolute ton of cells from this group are being called as negatives. And so what this means is they're unable to be assigned to a group, and so they're essentially being thrown out of downstream analysis steps, as is at the moment. And so that brings us onto our package demo for Demuxlet. Everyone with me so far? Good stuff. So let's see if our workshop is... Great stuff. So let's get started. We're going to load up a couple of different packages and find our color scheme for the heat map. So first of all, we're just going to do a little bit of like exploring data analysis to see what our data looks like, and then we'll get into the nuts and bolts of our method. So how's that going? Great. So basically, to motivate it, so we have lots of different hashing algorithms at the moment that just operate in hashing data. So with good quality data, they perform really well. With poor quality hashing data, performance reduces. So we can call some cells with hashing data, but not all of them. where are we going to start? Sorry, wrong page. So we have 3 data objects that we're going to load. We have a single cell experiment object, which contains RNA counts and HTO counts for a subset of one of our data sets we're working on. So it's roughly about 2,000 cells. As well as that, we have the corresponding SNP data for a subset of SNPs. And as well as that, we have a VCF file from the 1,000 genomes common variants that we're going to use to call SNPs on.
So wait, so you don't have to have called SNPs on your particular samples?
So calling SNPs is part of the workflow.
Okay, but you're pulling in known SNPs from the.
Yes. So you can call your own SNPs. So you can use something like Freebase maybe to call SNPs in your data. However, we're generally working on cancer data, so using known variants rather than just calling them in the data, it kind of reduces the chance of picking up a variant associated with like somatic mutation rather than something associated with the group.
Okay.
So yeah, we've this is just a sample subset. So let's get started. So our data set has 6 patients. So in this case, we had a mixture of our primary and metastatic groups, as well as this with some other covariates. So it's pretty important that we actually know which cells came from which person. We have our single cell experiment object, which the package integrates with, matrix of SNPs, and variant annotation, which is our SNPs in VCF format.
Did you make that, or is that like a publicly available?
The SNPs?
Yeah, the SNPs.
So there are lists of common variants. If you're interested, come up and chat to me at the end, and I can point you to those. So here, this is a subset, so it's about 2,500. If you look at 1,000 genomes common variants with frequency over, I think it's 5%, it's actually about 7 million SNPs. So to actually get the package onto Bioconductor, we've had to subset this. But one of the functions in the package actually subsets it for you. And I'll go through that in just a minute. So let's have a look at our hashing data. So as similar to some of the features I showed previously, we see some interesting things here. I'll point your direction to #2 here. So here we've actually very low signal altogether. So we don't know if this has got to be very few cells or because the hashtag just didn't take very well. If we look at #3 and 5, we see very low signal to noise ratio, quite a lot of overlap. As like a good example of what it should like, we can kind of look at #6 here, where we see pretty good separation between the two. So let's go down a little further. This is, it's going to come out okay, yeah. So this is when we apply strat HTO demux. I'll draw your attention to here at the bottom where we have this negative group. So we have 200 cells here being called as negative. This means that because of their hashing quality, they can't be called to a group and cannot be assigned. So, in total in this data set, we have 2,000 cells. So this is 10% of our data. That's a lot of our data chucking away, right? And we've anecdotally heard of different labs which have had similar issues. So let's look a little bit further into these. First, we'll look at the HTO, the sort of library size for the HTO counts, which we have here. So doublets, okay, much higher. That's what we expect. #1, 2, 3, 4, 5, 6. A little bit of variance. Then for the negative group, we can see clearly much more counts for the hashtag in the negative group. And that's kind of to be expected because that's how we define a negative. However, if we then look at the RNA counts, we see a different picture. So again, we see doublets, very high library size, as to be expected. 1, 2, 3, 4, 5, 6, very similar library size, but also the negative, very similar library size. So this indicates that RNA-wise, these cells are fine. We just couldn't assign these because they're hashtags. So these are useful cells. Right, let's keep on going. So again, as some like exploratory analysis, we can apply a couple of different algorithms, see how the results line up. So how is this coming out? That's okay. all on the same side. So here we're comparing site views and the strat H2 demux, which are two of the different algorithms. We see reasonable agreement, but again, we have this issue with the negatives in the strat group, and we see a lot of multiplets being called by site views. So in theory, we could take a consensus here to be confident that what we're calling is correct. However, that'll result in us throwing out even more cells, right? So we want to be able to reassign these somehow. So now we're getting into the functions in our package. So the first function we're going to come across is to pick out the top genes, and you'll see why this is important later on. It reduces the number of SNPs we need to look at, and as well as that, it reduces the noise in the data to some extent. So what this does is it looks at the RNA counts. It says which genes are expressed in almost all of the cells or most of the cells in this data, and then you can choose how many genes you want to return. So I shall run this here. So usually what we see here are the kind of housekeeping genes that are expressed in almost every cell. Yep.
I want to talk to you later, but do you think generally that is a good strategy if you're just trying to demultiplex from SNPs alone?
So
Or does...
We've found it useful, and hopefully again it will become apparent as I come down. So what we saw with some other algorithms that just operate in the SNPs data, and again I'm happy to go into this in more detail later on, is they're unsupervised. So they're looking for signals in the data, and when there was a large mismatch in size between the different groups, sometimes they could pick up different cell types instead of different groups. Because we're looking at mRNA, we see different genes being expressed in different cells. And so this goes some way towards mitigating that. It's something that we've yet to formally benchmark, but we expect by doing this, it somewhat reduces the cell type bias.
Okay.
So next we're going to subset the VCF. So as I was saying, in 1,000 genomes common variants, you could have up to 7 million SNPs, and we want to reduce this somewhat. So here, ours is in 7 million because we couldn't fit this on the package data. So we're just subsetting from like 2,500 to maybe 2,400 kind of thing. And let me run that here. So that runs too quickly.
Sorry, how do you subset? How do you choose which ones to?
Sorry, yes, good question. So the first function I showed you looked for the top genes in the data set, so the genes that were expressed in almost every cell. So that's fed into this function. So it subsets the 1,000 genomes common variants file to SNPs seen in those genes. So it excludes genes that we don't see expressed in most of the data. So that's our first filtering step.
Thank you.
So the next step here is when we want to start looking at reassignment. I'm kind of skip a step, but we can come back to that. And it's coming back to the hashing data. So we want to figure out which cells using the hashing data that we can confidently call. So this wraps around a package that I like called demuxMix, which is a mixture regression model, I believe, for operating on H2 data. But it's probabilistic, so we can assign a confidence score that a cell is indeed from a particular group. And so by assigning a high threshold, the cells that we get back, we can be quite confident they are from this group. The flip side of that is we end up with a lot of uncertain on cells, as we shall hopefully see here. So we've 608, I believe, uncertain cells. So the cells in hashtag 1, 2, 3, 4, 5, 6, quite confident they're being assigned correctly. And now we have this uncertain group. So the next step is the variant calling. So this generates our SNP data that our algorithm operates on. So we suggest using Vartrix. So it's a 10x genomics tool which calls SNPs in single cell RNA or DNA data. So what goes into this is your BAM file, your barcodes, your VCF file that we've just subsetted a minute ago. And this will give you a cells by SNPs matrix, which is part of the data included in the package. So you can follow these steps for your own data to generate the SNPs by cells matrix associated with your data. So now we want to look at reassignment. So we have the SNPs data now come out of Vartrix, and we want to stash this somewhere. So we're currently storing it in the alternative experiment slot of our single cell experiment object to keep everything nice and tidy. And so here we can see we've done a further filtering step. So this removes further SNPs that haven't actually been seen in our patients. So now we're down to about 85 SNPs in our alternative experiment object. And these are the SNPs that we'll use for our reclassification. So we've gone from about 7 million down to about 85 or so. So we can now visualize things, which is always good. So if we bring up a heat map of this, and hopefully this won't take too long, we can actually start to see the SNP profile associated with each distinct group. And what we'd expect to see is, in the singlet groups, a homogeneous SNP profile within each group. And hopefully this will run pretty quickly. So what we're going to see in a minute, once this finishes loading, is we have this negative group, which is about 10%. Sorry? Oh, sorry. Oh dear. The joys of live coding. Condition is length 0. Okay, what have I forgotten to run above? Oh, heat map. High confidence calls, I would say that.
While that's going on, if anybody has any questions or anything like that, we can help.
Would this be applicable to self-plexing? Not just HTO, but the new plexing from 10x, self-plex?
So I haven't directly worked on self-plex, but I understand it's quite similar in that you get this sort of like the hashing distributions. I would suspect it should translate across to that, but I'm happy to talk to you about it afterwards just to confirm.
Yeah. You've gotten self-plex to work.
There's been a few experiments run in my institute with self-plex. I think we lose about 20% of the cells because they can't be called. So I was wondering if
This is the same, right? I was talking to one of the cores here locally, and they were saying for BioLegend HTO types they're losing 20%.
Yeah, between 10 and 20.
And that's what we were seeing in this data. Like we were losing 10 or 20% with low recovery rate on some of the really important tumor samples, which is why we started on this whole thing of trying to see if we could use SNPs to recover those cells, because 10 or 20% when they're important cells, and those are by like patient tumor samples we'll never get back again. It's a lot. If it's cell lines, you don't care so much, but if it's like patient samples, we care.
So that I'm not wasting your very precious time in trying to figure out which code block I forgot to run, this is the heat map that we'll get out here. So we see this negative group across here. We have SNP data. It's not like all like a turquoise for no reads. So like, as we kind of supposed earlier, these are high quality cells. And so if you kind of look across, most of the negatives look pretty similar, right? And if you look pretty closely, they actually look quite similar to the #5 group. So we kind of could suppose that they might go in here, but obviously we want to kind of do this in a slightly more systematic way rather than just like manually moving cells, right? So we have our reassignment algorithm, which unfortunately I can't run for you because I seem to be missing something, but it uses a k-nearest neighbor model. So it takes the singlets that we can call with high confidence. That's your training data. So we are very confident of the labels of these. We then simulate some doublets using these high confidence singlets. So we now have a good training data set that we know that what the singlets are, we know what the doublets are. We then predict these on the remainder of the cells that we couldn't confidently call using the hashing algorithms because we know the SNPs associated with them. So for example here, this is a subset of data. So this is the Seurat H2 Demux negative group, just the negative group, right? So these are the cells that we previously couldn't call. And we now, I got that wrong. Sorry, the new #5 group, yeah. So this is the #5 group called by our algorithm. So as we can see here, we have cells that were initially called as #5. We now have the negatives that we kind of presumed look like #5. And we also picked up some other cells from other groups. And you can see looking across here left to right, we have a really consistent SNP pattern. So we can be quite confident these are indeed cells from the same person. So I'll do some like very quick benchmarking. So on this, we've done some more form of benchmarking since, but we can just isolate the high confidence singlets. And from this, we can split into like a training and test, predict, and we can see quite good agreement between the two. Also, which is a feature that I quite like, if I can find it here. So basically in our training data, we're kind of assuming these are the cells we can call with high confidence, right? But what if there's some errors in the training data? So we can actually like intentionally mislabel some of these. So here I've taken 25 cells here from #5 and renamed them as #2. So then if we predict the model back on itself, we can actually see that they are now correctly relabeled, showing that we have some level of robustness to errors in the trained data. So obviously in practice, this is like a very, very, very basic level of benchmarking. We've presented some of our more formal benchmarking at ISNB last week, and I'll be happy to share that with anyone who's interested on like simulated doublets, more realistic data. And we do see that it increases the performance of the hashing methods where we have poor quality hashing data. It matches the SNPs methods like Supercell and Vireo on kind of like a normal data set. And we do suspect that we will have gains, for example, when we have very imbalanced group sizes. Here, kind of anecdotally, we can kind of see this is our demuxSNP #6 group split by the demultiplex classification. And again, we can kind of see if we look at this like one cell which was called differently, and we zoom in, we can kind of see it probably does look a little bit more like a #6. So this does provide some way of QCing. So to summarize, using this method, we can identify cells which have been miscalled or uncalled by their hashing algorithms. And as well as this, we can reassign cells which have been miscalled or uncalled by other hashing algorithms. So just to wrap up, I'd like to say a big thanks to the support from my supervisor, Professor Aedin Culhane, my co-supervisor, Laurent Gatto, as well as the rest of our group at the Culhane lab. Thank you very much. I'd be happy to discuss this and take any questions from people.
Any questions? I have a question. I have some samples that I hashtag with like a healthy donor with a tumor. So they would be like within the same hashtag, have two different, like from two different people. So looking at like immune response. Would that confuse the algorithm, or would it be able to handle that like #1 could have two different SNP profiles?
So the two samples, they're from the same person?
No.
Oh yeah, then it's good to go, yeah. Once the two or however many samples you have are genetically distinct, then it should be good to go.
Yeah,
Yes.
related to that, I mean, have you tried, if you have like a tumor normal sample from the same person, can you use just a small number of like somatic mutations in the tumor to distinguish it? Like what's the number of SNPs or variants that you need to be able to distinguish them?
That's interesting. So at the moment, we've just applied it to data where we're looking at different individuals.
Yeah.
And again, we're using common variants. However, if you were to do that, I guess it depends on how consistently you see that mutation coming up and whether that's a strong enough signal to distinguish the two. Definitely will be an interesting use case and be something for us to look into for sure. Yeah.
Stephanie Higgs published a paper as well trying to call somatic mutations in tumor cells.
Mm-hmm.
So that could maybe look at the mutation rate or the estimated mutation rate across cells. I suspect if you wanted to, say that these are somatic mutations in the epithelial tumor versus the normal cells, but then it might be confounded a little bit because I think some of the recent studies where they've looked at normal cells around tumor cells can also have a lot of somatic mutations. Mm-hmm. So I suppose it depends on the distribution of those somatic mutations.
I think you definitely could probe the data a little bit better using this. But again, it would come back to how many SNPs you had. If it's just two samples, it'd be a little bit easier. As you increase the number of samples, you kind of need more SNPs to separate the data, but it'd have to be an interesting application. Yes?
So I have a question do you find that in practice, the demultiplexing quality is related to the data, the scRNAseq data quality?
So it may be, but not necessarily in every case. So it's not something we've formally tested, but if I was to speculate, if I can bring up another plot here. So if we look at this plot here, right? This is the HTO library set. And here we see very large differences between each of the groups. However, looking at the RNA library size, it looks to be a lot more consistent. So there may be an impact by the RNA quality. However, I don't think that's the only factor coming into play.
Oh, I see.
For example, like these are all sequenced together, so in theory they should all have the same sequencing depth. I know some models have actually scaled by that as well, and it does seem to improve performance. But I suspect there are other factors coming into play as well.
I have one more question. Thank you very much for the workshop. If we're working with different patients, should we try to use HTOs or should we just go for SNPs?
I would be very much pro using the HTOs. The HTOs aren't perfect, and in our lab and in some other labs, we've anecdotally heard of issues with the tagging. However, this is why I'm quite pro HTOs. So we applied some of the SNPs algorithms when we saw we dubious HTO quality. And what we got was, so if I go back to my hashing distributions, so in this #2 group here, for some unknown reason, we were unable to recover very few cells. We loaded equal numbers of cells for each of the 6 groups. For some reason, the #2 group, we lost a load of cells. When we ran Supercell and Vireo, in hindsight, we suspect because this was quite a small group, they actually missed this group entirely. So in our original data set, we had about 20,000 cells. Supercell produced a #2 group with about 1,000 cells. But in analyzing it after the fact and looking at the SNPs, we could quite clearly see this was actually very much a doublet group. And we wouldn't have known that without the HTO tags. There would have been no way to cross-check, which is why I'd still be pro the HTO tags. As well as that, to the best of my knowledge, unless you have genotype information with the SNPs algorithms, you can't actually match it back to a patient. It'll split into groups. However, you won't know which group came from which person without the genotype information. If you do have the genotype information, then great.
Thank you.
I think there was also a good bit of disagreement in our data between Supercell and Vireo,
Yes.
which was kind of surprising. So then which one do you trust?
Any other questions? Do you find any particular hashtag to underperform in comparison to others? I mean, here #2 that didn't seem to work. Is that always the case, or do you think that was just an experimental?
We would suspect. So we don't think the issue here is with the hashtag itself. It's something within the experiment that we lost a lot of cells from #2. We initially suspected that all those negatives might have been from #2. However, again, looking back in hindsight and analysing the data, we can be quite confident they are indeed from #5. So we just suspect that for reasons unrelated to the actual hashing itself, we just lost a lot of cells from this group.
So, how many have you run successfully on how many different experimental data sets?
So this is obviously a real data set. We don't have ground truth.
Yeah.
So what we're kind of seeing here, where it's a little bit of qualitative. However, we are benchmarking. So we've taken like the high confidence singlets, simulating doublets from that. Again, happy to go through this in more detail at the time. We see good performance on the sort of data set we've simulated from this with good ground truth. We've run it on some PBMC's data set with much lower sequencing depth and fewer cells. We see a slight reduction in performance, but still, if you have poor hashing quality, it will improve the performance. And so we're kind of looking to expand this out now. It is also on the Bioconductor development branch if anyone's interested in having a look. Yes.
I wonder if you could combine this with like CRISPR, like for CRISPR perturbations in single cell RNA. If you use like a cutting site, like for like some of the CRISPR experiments, combined with like a hashtag. It's the same person throughout the different hashtags, but different CRISPR conditions.
If you have a strong signal there, then you potentially could. It's not something I know a massive amount about. That's having something we've actually tried, but it's a possibility, yes.
If there are no more questions, you can thank Michael.